Whenever you're ready.
Okay, so should we experiment on the human genome? So clustered regulatory interface, short palindromic repeats, also known as CRISPR, is a new way to genetically modify genes safely and efficiently. More accurate than other methods, it has the ability to add, alter, and delete different genes. It has been determined superior to other methods because it's cheaper and a lot more customizable. It also has the ability to secrete its own enzymes such as Cas9, which is also known as molecular scissors. Its job is to locate and cut strands of DNA at very specific locations. This has really helped shape the CRISPR technique. The CRISPR-Cas9 system has the potential to cure genetic diseases and edit future offspring. There are many discoveries that have been made throughout history with thousands of scientists in different communities. The advantages of the technology include the ability to edit somatic and germ cells, not one or just the other. It also needs regulations, but that's also part of coming solutions.
Yes, we should experiment on the human genome. This opens the door for many possible uses, including curing genetic diseases. Pros of CRISPR-Cas9 and human genome engineering are we could completely eliminate genetic diseases, both fatal and non-fatal, and it would give parents a chance to have a healthy child. Some medical uses of CRISPR are it can be used on patients to cure genetic diseases. This would give parents who suffer from disease the opportunity to have a healthy child. Genetic disease would be cured for good since genes are passed down through offspring. CRISPR can also be used on plants to create enhanced crops that are immune to disease, higher in nutrients, and can adapt to harsh environments. Human genome engineering could also be used for enhancement purposes instead of medical, which would be detrimental to our society. Some cons of CRISPR-Cas9 and human genome engineering are enhancement procedure would most likely be expensive, which would expand the divide between the rich and the poor. Another con is groups with traits seen as undesirable could be marginalized. Minorities or people who don't fit the high beauty standard could face severe discrimination. Some uses of enhancement are that CRISPR could be used to control superficial traits like eye color, hair color, height, intelligence, sex, or athletic ability instead of curing genetic disease.
And how can we make this possible? There are two medicinal uses of CRISPR, somatic cell therapy and germline cell therapy. In somatic cell therapy, a sample of cells is taken from a patient, modified using CRISPR, and then reimplanted. Similar processes are being used to treat leukemia, sickle cell anemia, and other blood-related diseases. In germline therapy, the sperm and egg cells are modified to eliminate all the DNA sequences that code for genetic disease. Then the zygote is formed in vitro and implanted into either a mother or a surrogate.
So a little history and politics of, were you done?
No. To prevent exclusion, elitism, and elitism, treatments must be made accessible to all social classes. This technology must also be regulated to prevent any misuses. Similar regulations have been made in genetically modified crops. In the U.S., genetically modified crops are regulated by the Environmental Protection Authority, U.S. Department of Agriculture, and the Food and Drug Administration. These agencies address the effects of bioengineered crops on the environment, agriculture, and human health. Laws and regulatory bodies must be placed on the human applications of CRISPR-Cas9 to eliminate all possibilities of misuse.
Now a little history and politics. So CRISPR was announced in 1987. There have been lots of historical breakthroughs throughout the years. More specifically, one recently in 2020 with the Nobel Peace Prize awarded to Emmanuel Carpenter and Jennifer Doudna. And many scientists have been using the technology across the world. More specifically in France in 2005, it was discovered that CRISPR can provide cell immunity for genetic diseases, which was a huge breakthrough. And more later in 2013, another big discovery was that CRISPR-Cas9 could be used to modify plants, tobacco, rice, and wheat, and also zebrafish and rats, which was another huge breakthrough because it shows a large step in being able to genetically modify animals. And research and development continues to be pursued even in 2020 with COVID. More recently, in March, genome editing therapy was directly administered to a patient in the body, allowing for continued growth and development of the technology. The The Silmar Conference was also a large step in the process. It was one of the first discussions about issues and successes in the political category of human genome editing. It took place in 1975 and consisted of biologists proposing a moratorium on germline genome editing and recommended guidelines. Regulations and limits to reach. It also discussed regulations and limits to reach without endangering public health, as well as address risks of editing genomes and making sure it isn't a poor model for other methods. It brought up environmental issues such as unleashing dangerous organisms into the lab, and have the possibility of injuring humans or ecosystems. It still took decades to build any consensus on how genetic development should be governed. The presidential elections have been helping with the political side of human genome editing as well. Presidents are getting involved in processes by creating policies and regarding the discoveries. And this led to anxiety among scientists, but also allowed for a lot of political research to be taken from the political leadership to be taken from the presidents. And in the past 3 presidential terms, there have been heated debates about ethics of cloning and embryonic stem cell research. And this has allowed a lot of controversy, but also a lot of progress. And biomedicine has achieved high political standards and acceptance throughout the years. And President Bush played a really key role as he emphasized public reflection on ethical issues rather than policy recommendations. So he really looked at the bigger picture instead of just the small political issues.
The use of CRISPR-Cas9 on human embryos has sparked an international debate between scientists and regulatory bodies. In November of 2018, at the International Summit of Human Genome Editing in Hong Kong, Chinese scientist Zhang He presented his experiment, which caused worldwide uproar. He used CRISPR-Cas9 to create genetically modified babies, which were immune to HIV. In response, the Scientific Ethics Committee of the Chinese Academy of Science declared their opposition to the use of gene editing technology on human embryos. They claimed that it was in violation of laws, regulations, ethical norms, and without full scientific evaluation. Human genome engineering could take a turn towards eugenics, which is the selection and glorification of those traits seen as desirable or superior, and the marginalization of those traits seen as undesirable or inferior. This could lead to the discrimination of those minorities that don't fit the societal beauty standard. Considering high racial tensions and past uses of eugenics, this is a possibility to be wary of. While CRISPR is an effective medical treatment on the somatic cells of consenting and sentient patients, its application on human embryos poses too many ethical violations for it to be a viable option. There are too many risks and unforeseen consequences for this technique to be a common clinical practice.
Futuristic lens. Using all of the research on the human genome that scientists have done over the years, researchers now know the DNA errors that happen to create over 6,000 diseases, and scientists can use CRISPR to fix those errors. With the power of curing diseases, it could be good if it's used correctly. Researchers suggest that there should be some types of restrictions put on this technology so that the power can't be used in a way that could lead to the downfall in the future. Another way this technology could be used in the future is to create designer babies. A designer baby is a baby genetically engineered in vitro for specific types of traits. With the production of designer babies, there's a possibility to create a child with a DNA sequence that the parents pick out. Therefore, parents could make sure their child doesn't suffer from hereditary disease that is carried by one of the parents, while also picking out the child's hair color, eye color, skin color, etc. Changing the genetic code of organisms creates many unknowns for the future. There are scientists working on editing the genetic code of animals and then having those changes passed down to their offspring, eventually creating a whole new genetically modified population. Lots of scientists' work has been focusing on eliminating Lyme's disease from the population. While CRISPR could bring us huge benefits for the future, the stakes are high. Even with all this development and research that is done, scientists are working on being able to undo what they have done, just in case it goes wrong. Until the undo is complete, scientists urge people not to use any part of the creation yet.
CRISPR is like a double-edged sword. While it has promising potential to cure genetic disease, it could also be used to unethically enhance human embryos, which could cause a plethora of societal problems. Effective regulation was placed on genetically modified crops in the 1980s. The potential consequences of genetically modified humans outweigh the consequences of genetically modified crops, but the administrative framework the 1980s regulations left behind could apply to the current dilemma. Hopefully, after much discussion of the safety, ethical, and scientific concerns of the human applications of CRISPR-Cas9, similar restrictions and regulatory bodies will be put in place to safeguard society, eliminate abuses of the technology, and put to rest all detrimental outcomes.
Okay, so some questions here I have for you. Emma,
Yes.
describe how the content of the team presentation was changed as a result of your discussions as a group.
As a group, we were really able to discuss each of our perspectives together and bring all that information together. So like when we first sat down, we all kind of talked about our own papers, we all shared our essays, and we all just kind of collaborated on that. And it brought information that helped our argument just really come into place and really have different perspectives to bring into the argument and really prove our points.
Okay. Natalie, give one specific way that your thinking changed as a result of Clara's findings.
So at first, I was kind of leaning towards the no. Like, should we, like, in my paper, I was starting to, like, lean towards saying no, should we experiment on human genome? But reading over these two papers, they both shaped, like, and changed perspectives in my brain. Just looking forward to, like, all the diseases that could be created or, like, cured, and just how it can enhance so many, like, possibilities in the future. Yeah.
Okay, thank you. Clara, reflecting on your colleagues' work, which one had the greatest impact on your overall understanding of the problem your group identified?
I think that Natalie's futuristic lens helped a lot with like the whole, like the worst case scenario, and that helped with my ethical lens of how that worst case scenario affects like society. Like she went into a lot of like the designer babies and how like a genetically modified population would be harmful to society, and that was really useful information to me because that was an area of my research that was kind of weaker, and so I was able to apply her research to my own.
Okay, Ruby, thanks. Hey, another round of applause.